Hello everyone, am I audible and visible? So, last class we discussed about the transplant statistics and what do the hospitals do when it comes to organ transplantation. And we also discussed the importance of genetics in organ transplantation. And we finally moved to the stem cells and its types. And we concluded the session with the protocols for creating iPSCs. So, in today's session, we'll be discussing how the diseases are modeled conventionally. Basically, we'll be discussing about animal models and computational models. And then we'll be finally jumping to the hero of our module, which is organoids, and how they help in disease modeling and drug testing. And then I'll be teaching you the 3D bioprinting. And then we'll have a guest to teach you about to demonstrate how to use the software Fusion 360. So, are you guys ready? Give me a plus one or a thumbs up. Guys, that's awesome. So, let's get started. So, how are diseases modeled? Let us first discuss the conventional methods. The first method is animal models. Scientists use animal models because they offer a complex biological system that allows them to study the disease development and to test potential treatments, and they gain valuable insights that wouldn't be possible with simpler models like cell culture. So here there are two categories. The first one is spontaneous models where the animals develop the diseases naturally. Examples are diabetic mice and beagle dogs. The second kind is induced models, where the diseases are artificially induced in healthy animals through various techniques, such as genetic engineering. Here, the scientists introduce specific mutations or genes associated with human diseases into animal genomes, which creates a knockout or overexpressing model. So, what do we mean by a knockout model? We completely suppress a particular gene from expressing. So, Arun also has discussed about this technique in his session 3. So, let's move on to the second process, which is chemical exposure, where the animals are exposed to chemicals that trigger a disease process, such as exposing rats to carcinogens to study cancer. The third process is pathogen infection, where the animals are infected with specific pathogens such as bacteria or virus that cause similar diseases in humans. So the example is mice infected with influenza virus to study the flu. Now, why is it not 100% successful? There are limitations for it. So let us see why. Uh, the animals and humans have different physiologies and metabolisms. So the drugs that work well in animals may not be effective in humans. So, for example, a drug targeting a specific receptor in mice might not have the same effect on the human version of that receptor. And these animal models often fail to capture the complexities of human diseases, which can lead to wasted resources and failed clinical trials when the drugs appeared promising in animals does not translate the same to human patients. So, do you guys know what actually happens when it comes to animal testing? Any ideas? What do they do to that animal? Not really. Okay. So let me explain that to you. Let's say we have, yeah, but how do they observe? So uh, what happens is, let's say there are two mouse, mouse A and mouse B. Let's say the mouse A eats a lot and mouse B doesn't eat as much as mouse A. So we infected both the mouse with a particular disease, and after 1 day of infection, we kill the mouse A. So this is what actually happens when we do animal testing. We kill that animal and then we take the proteins from its body to test it to see what actually happens in the body of the mouse. So, for an example, when we observe the mouse A, there was a lot of glycogen present in its body, which is 1 day after the infection. Now, after the second day, we kill the second mouse as well, and when we tested it, we noticed that there is a less amount of glycogen as compared to the mouse A. But here the problem is we cannot conclude the variation in glycogen, whether it's because of the number of days of infection or the dietary habits of that mouse. So this is an example where the test doesn't actually go well, or it's not that accurate. And this is very cruel as well, which raises ethical considerations because the animals also experience pain and suffering and psychological distress such as fear and anxiety, and they also deserve moral considerations similar to human beings. So, did you guys get this concept? Okay, nice. So, one example of a drug that passed the animal testing but failed in human trials is the fialuridin drug. It was designed to treat hepatitis B, uh, and it showed a great promise during the testing on mice, rats, dogs, and even primates. It effectively reduced the amount of hepatitis virus in these animals. But during human trials, there was a very bad side effect happen, and 7 people developed liver failure, and even 5 people died of it. So this development was halted because of the unforeseen risks which happen in human beings. So this is the reason why animal models are not 100% efficient. Now let us move on to the next model, which is computational modeling. They offer a powerful tool alongside the traditional methods like cell cultures and animal models for understanding and predicting diseases. So let us see how they play a crucial role in disease modeling. They help in simulating complex systems. Uh, Unlike the physical models, the computational models can represent complex biological systems, including the interactions between genes, proteins, cells, and organs. So, this is not very easy to study in animal or cell cultures. And these models can simulate how diseases arises and progress by using a vast amount of biological data. So, what do we do here? By manipulating the variables within the model, we can predict how a disease might spread or how it might respond to different interventions such as new drugs or vaccines. This allows for in silico, which means computer based experimentation, which saves time and resources as compared to the traditional methods. Here, the models can also be used to compare the effectiveness of different treatment options and predict how a patient might respond to a specific therapy based on the individual genetic makeup. This personalized approach to medicine is becoming increasingly important, and we'll be discussing how the lab grown organs helps in personalized medicine in the next section. So I think this is the right time to give a brief introduction about our club's projects. The first project is autism modeling. This project we are doing this year, and in this project we are working to create a model for biological motion processing of an autistic mind. Here we create a model similar to human brain, and we damage this neural network to obtain the sensory output an autistic person would receive. So what is the output of this project? We can come up with novel therapies and even a cure for autism. The second project is AIS, which is Artificial Immune System, which we did last year. So this is a bio-inspired algorithm, which is negative selection algorithm to detect Parkinson's disease from a patient's handwriting. Early detection is very crucial for diseases such as Parkinson's because if therapy is started early, we can slow down the progression of the disease. Now, why do we use negative selection algorithm here? So for that, we need to know what happens normally. How do we test this disease normally? There are two other algorithms, which is KNN and SVM. Uh, these what happens in KNN? Why it's not feasible for everyone is KNN is a very expensive process, and for SVM, we need equal data for healthy and unhealthy population. We need equal data set, but right now we don't have that much data when it comes to unhealthy people. So this won't work. So to create uh, a solution for these problems, we created something called as anomaly detection, which is nothing but negative selection algorithm. What happens in this is we have a data set for healthy population. Let's say a patient or a person gives his or her sample. When this sample, if this sample matches, if the data of the sample matches the data which we have with us, we can conclude that that person is safe from Parkinson's disease. But if that data does not match with our present data, we can tag that person as a red flag, and we can say that that person might have Parkinson's disease. Then after this test, they can go for those expensive processes to for confirmation. So, so we saw that this is a bio-inspired algorithm, right? So how is it bio-inspired? So as we discussed last class, our immune system also works in the same way. So we discussed about it when we were discussing about HLA matching and blood typing as well. So what happens? Our immune system can differentiate between self and non-self. If it sees a cell from our own body, it does not do anything to it. But if it identifies a foreign cell, what it does is it goes and kills it. So this is the uh, biology where we took our inspiration from, and our model works very similar to our immune system. So do you guys have any doubt in this? Can we move on to our next topic? Okay. So now let's just one. Thank you, Gaurija. Now let's move on to discuss about the hero of our module, which is our organoids. So, what are organoids? How are organoids useful for scientists and how they reduce the use of animal models? So, let's deep dive into this topic. Imagine tiny, simplified versions of organs grown in a lab dish, such as a petri dish. These are called as organoids, like little balls of cells that acts as a simplified organs. They are built from stem cells, uh, which we discussed that those amazing cells can become into any cell type. Now, what are the benefits of organoids? So, the major benefits of these organoids is in drug testing and disease modeling. So, let us discuss the benefits in drug testing first. Think of organoids as disease playground. Here, scientists can grow organoids that mimic specific organs like a mini brain or a mini gut. This is very important because scientists cannot try new treatments on patients. So by introducing different diseases into these organoids, researchers can see how the disease affects the cells and the tissues, just like a tiny petri dish version of what happens inside our body. So, this helps scientists understand how the diseases work, which is a very crucial step in developing treatments and cure. This also this also helps in improved drug selection because with a more human relevant platform, organoid testing can identify promising drugs with better efficacy in human beings. It also has the reduced risk of failure because drugs which shows negative effects in organoids, uh, like failing to interact with the disease target or causing unexpected harm, are less likely to be effective or safe in humans. So, identifying these issues early on saves time and resources that would have been spent on the later stages of clinical trials. The organoids are very important for personalized medicine as well. So imagine treating a group of people with allergies using the same medication. It might help some people, but other people might have no response to it or may even experience side effects. So similarly, the traditional drug testing doesn't account for individual variations. But how does organoids help us here? Uh, Organoids can be grown from a patient's own cell, right? We discussed it in last class also. So, which creates a miniaturized version of the organs with the patients with their unique genetic makeup. So, we can uh, we can create medicine depending on the patient's genetic makeup, and most likely it will be a success in that patient. Now let's discuss about the disease modeling aspect of organoids. For diseases with strong genetic component, patient derived organoids can be created from the diseased tissues harboring the same genetic abnormalities present in the patient. So this allows testing the drugs on the cellular model that closely resembles the patient specific disease. And by studying the organoids derived from patient with specific diseases, Researchers can gain valuable insight into disease mechanisms. So, uh, what do they do? This can lead to a deeper understanding of the underlying cause of the disease. So, scientists can study what causes the disease so that can lead them to create a better treatment option. So, analyzing the difference between a healthy and a diseased organoid helps researchers to identify the key pathways and genes involved in the disease development. So, it leads to a better and effective treatments as well. So, this also paves a way for personalized treatments. So, let's talk about a real life example where organoids were used. Let's talk about brain organoids. So, our neuroscientists face a challenge because our brain is shielded with thick skulls and layers of protective tissues, and human brain is extremely difficult to observe in action. And for centuries, our scientists have tried to understand them using the autopsies and animal models. And in recent years, they tried to understand the human brain with imaging techniques as well. We have learned a lot through it, but there are still many limitations to it because conditions like Alzheimer's and the effect of human brain and the effect of these diseases on human brains, such as Zika, continue to hide beyond our view and our understanding. So, how do we use organoids here? So, organoids were used in uh, treatment for a disease called as microcephaly, which is a disease caused by Zika virus. So, do you know what happens if a person has microcephaly? Any idea? You can respond in the chat. Yes, it is right. So let me explain it to the people who doesn't know about it. So microcephaly is a birth defect where the baby's head is smaller than expected. It can lead to developmental issues as well. And scientists use brain organoids derived from healthy cells and the cells with mutations linked to microcephaly. By studying these miniature brains, researchers were able to identify how the Zika virus disrupts the brain development in microcephaly cases. This finding helped the scientists to understand the disease process better, paving the way for potential therapies. So they found two drugs, which is pipestrine hydrobromide and amodiaquine dihydrochloride dihydrate. These are two drugs which were found to be effective in eliminating Zika virus in human pluripotent cells, which are derived from brain cells. It is a type of brain cells. They tested these drugs on a type of cells called cortical neural progenitor cells. So, do you guys have any doubts here? That's about organoids. Could you please? Yeah, I'll type it here. So, are you guys ready to jump into 3D printing? Give me a thumbs up, guys. I see only a very few response. So, what I'll do is first I'll explain you what is 3D printing and what is 3D bioprinting, and then our guest will teach you how to model it. But it will be better if you listen to what she's teaching, and you can try out later after the session because it's a bit hard, and so no need to do simultaneously. We'll provide you the links where you can learn more about 3D modeling as well. So no need to worry about it. Is it clear, Rico? Okay, so let's get started with 3D printing. 3D printing is also known as additive manufacturing, and it's like a fancy printer for creating real objects. So what happens in it? The first process is we design it. We start with a digital 3D model of the object which we want to create. So this can be done with a specific or special software such as Fusion 360 or Blender. We'll be using Fusion 360 today. And the second process is slicing, where we use a software to slice the 3D model into many thin layers, like slicing the bread. The third process will be printing it. The 3D printer reads the instructions from the sliced model and builds the object one layer at a time. And it uses various techniques depending on the printer type, but it generally lays down a thin layer of material, then hardens it with UV light or heat, then it moves on to the next layer. And this process is known as cross-linking. So imagine building a brick wall one at a time, but instead of bricks, we used plastic or metal or even concrete depending on the type of printer used. So these are the name of most commonly used 3D printers, which is Prusa Research or Bamboo Lab. So why is it very useful? What are the benefits of 3D printing? The first thing is less waste. Unlike our traditional manufacturing where we carve an object out of a bigger piece, 3D printing uses only the material needed, which significantly reduces the waste. And we can create complex shapes that would be very difficult or even impossible to create with traditional methods. The third one is customization, where diseases can be easily modified for customization or for creating different prototypes. So let's apply the same concept in 3D bioprinting. So what happens here? Uh, Imagine a high-tech printer, and instead of ink cartridges, we use inks which is made of living cells, which is nothing but our stem cells. And let us see how does it work. So here, the first process is printing the blueprint or designing. The scientists use a computer to design the 3D model of the target organ, like a tiny heart or a thin sheet of skin. So this blueprint tells the bioprinter where to put everything. The second process is squirting the bio ink. So this bioprinter is like a high-tech chef uh, bio following the blueprint. It squirts out various inks containing different types of stem cells. So, for example, in one inkjet, there are stem cells for organs and stem cells for blood vessels. So we can print using different stem cells at the same time. And these materials are put layer by layer precisely where they need to be to mimic the natural structure of the organ. But let me ask you a question. The ink is a liquid, right? We put it in a liquid form, but the final organ which we get is in a solid form. Now, how does this how is this possible? Do you guys have any idea about it? No. Okay. So, do you guys know? Uh, Not fully true, Jyotin. But do you guys know about SDS page? Let me explain it to you. SDS page. Okay. So some people is saying no. So I'll explain it using both the methods. So we create jellies. We eat jellies, right? So during the manufacturing, they put it as a liquid, and with the process called as polymerization and cross-linking, which we discussed in the 3D printing introduction part as well. During that, using that process, the jelly solidifies. So if I have to explain it using SGS page, we add something called as TMED. Do you guys remember it? We add something called as TMED, which is a solidifying agent for the gel. So what happens? It cross-links the acrylamide present in the gel. And finally, after 15 to 20 minutes, it solidifies, right? So, and but here we can use that process as well. Here we use UV light to solidify the ink which we use. But we created the structure, but how do we tell the stem cells to grow in that particular way in which organ we need them to grow? How do we tell the stem cells to grow as our requirement? What do we use? Anybody? We discussed about it yesterday in the last class as well. We use iPSCs. No, no. How do we tell stem cells to grow in a particular way? Scaffolds. It is for the scaffolds are used for the uh, like a bag like a backbone. It is used for the structural support. But functionally, how do we make the stem cells to grow? Okay, let me tell. Yeah, growth factors. I was expecting this answer. We use growth factors for, and after printing the structure, we give them growth factors so that if the structure is a heart, they can the stem cells can uh, perform the function of a heart. So we can grow them in that manner using growth factors. For example, there is growth factor EFG, for which is used to create epithelial uh, cells from stem cells. And there is a growth factor called as fibroblast 10, which is used for stomach, breast, and liver tissues. So, using these growth factors, we can convert stem cells into any organelles. Yeah, I'll share the PPTs. UV Barry. Our biotech club doesn't have a bioprinter, but we also have not done that before. Yeah, I'll share the images of. I'll share the images of 3D printed organ by the end of this session. Is it okay with you? Okay. So the third process is we once the stem cells are printed in the right places, they start to specialize and connect with each other, forming the building block of the desired tissue. Uh, how does this happen? By using our growth factors. And the fourth process is growing and plumbing. Bioprinting can also include instructions for incorporating channels within the printed structure. And these channels act as a mini highway, allowing the nutrients to flow through the growing organ, mimicking the role of a blood vessel. And this is called as vascularization. And it is crucial to keep the cells alive and functioning. Now, what is the material used for 3D bioprinting? Anyone? What is the material used for 3D bioprinting? So, tissues, stem cells. No, those are the main components of the bioink, but they need to be present in something, right? Like, uh, what do we say? They need to present in polymer chain. Yes, very similar to that. So, it is a gel-like material. Let me explain it to you. So, in 3D bioprinting, we use something called as a bioink. So, this we use to create structures layer by layer, and this bio ink needs to be very special because it carries live cells, and so this ink needs to be biocompatible as well. So what do we mean by biocompatibility? It should not cause any harm to the living cells. It should not be toxic, and that ink should support cell growth and function as well. So let us discuss the main types of bioinks. The first one is hydrogel. So these are gel-like materials often made from natural substances like collagen, alginate, or gelatin. They provide a scaffold for cells to uh, stick to and can be engineered with different properties to mimic the environment or specific tissues. So we discussed now what is a scaffold. We discussed about scaffold in last class as well. Scaffold is a 3D structure that acts as a supportive framework for cells. And scaffold provide a temporary structure to cells to stick to and grow on, and to organize the cells into a desired structure. So this scaffold also need to be of a biocompatible material. The second type of bioink is synthetic polymers. These are man-made materials like polyethylene glycol or fluoronic. They offer good printability and control over the structure of the bioprinted object. But in this case, biocompatibility is a concern. Uh, we are not sure whether it is safe for the cells to use when we use synthetic polymers. The third one is decellularized extracellular matrix. Here, the bio ink is derived from natural tissues by removing the cells, leaving behind the complex molecules that provide the structure and functional support. So, it offers advantages in terms of the biocompatibility, and also it mimics the natural tissue environment. So I'll show you the extracellular matrix of a heart by the end of the session. Now let us move on to our activity, which is 3D modeling of trachea. So what is a trachea? It is also known as the windpipe, and it is a tube-shaped organ which is present in our neck and the upper chest, from the neck to our upper chest, and that is very essential for breathing. So, what is the location of it? It starts at the bottom of our throat, just below our voice box, and it travels down our neck and chest until it's split into two smaller tubes called as bronchi, which leads to each lung. What is its function? Its main job is to allow air to travel between the nose and mouth and between our lungs. So, what is the structure of trachea? Trachea is a hollow tube made of many C-shaped cartilage rings. And what is the function of these rings? They help the trachea to keep it open, like they act like a support beams, preventing the trachea from collapsing, and it ensures smooth airflow as well. And this allows flexibility for the trachea as well, which is important for movements like turning our head. Trachea also has an inner lining uh, with a special tissue called as mucous membrane. So this lining has two important parts, which is mucus, which is a sticky substance that traps dirt and other particles from entering our lungs. The second part is called as cilia. Cilia is nothing but tiny hair-like structures that move back and forth, helping to sweep the mucus and trapped particles upward and out of the trachea. So I hope you understood the function of trachea. Do you guys have any doubts in this? Do you guys have any doubts so far? No doubts. That's great. So let us move on to our demonstration. So let us invite Diksha from Bioprinting Club. So she's a coordinator in not Bioprinting Club, sorry, it's 3D Printing Club. So she's a coordinator in 3D Printing Club, and she will be teaching you to use the software Fusion 360. So let us welcome her.
Okay, hi guys. So today we'll just learn on the basics of fusion. In 1 hour, I'll be teaching you some basics required to model the trachea. But we'll be assuming the trachea to be perfect cylinders because fusion doesn't allow us to model irregularities. And that can be modeled using Blender software. And we'll be looking into that in the next session. Okay, I'll share my screen. Okay, so I hope you guys can see my screen. So if you open Fusion, this is how your Fusion screen will be looking. So all the options here are basically your 3D options. And before using our 3D options, we'll be using our 2D options to create a sketch, and then use the 3D options to extrude, revolve, etc. So how do we create a sketch? So on the top you have all your options here, and the first option which says create sketch, you will have to select that. So when you select on that option, it will ask you the plane in which you want to create your sketch. So you can choose any plane. So let's say let's choose our top plane. So this is your top plane, this will be your front plane, and this is your side plane. And so you can select any plane. So when you click on the plane, this is how your screen will look. And so these are some of the options needed. So here under create, we have many options like line, rectangle, circle, arc, etc. So since we'll be modeling a trachea and we are assuming it to be a perfect cylinder, let's just draw a circle. So to draw a circle, under create, go to circle, and there are many options like 3-point circle, 2-point circle. For now, let's just use the center diameter circle. So if you click on that, your cursor will change into a plus sign. So it will ask you to select the center of the circle. So for now, let's assume the center to be at the center point itself. So once you have come to the center point, just uh, left click on your mouse and move it, and you'll be able to draw a circle of any diameter. So you'll have to mention the diameter of your circle. So in the text box there, just mention, let's say for now 60, just type in 60. And once you're done, just left click on your mouse again. So for Fusion, I will advise you to use mouse because it's easier if you're using a mouse. And it can be done without mouse, but it will be a little harder. So this is how you draw a circle. Now let's say once you're done, and if you don't want your circle command anymore, just click on escape. So like that, you can draw other shapes as well. Let's say if you're drawing a rectangle, so there are many options like 2-point rectangle, 3-point. So center rectangle is basically, it'll ask you to mention your center of the rectangle and you can extrude the rectangle. 2-point rectangle, basically you will have to mention any of the two vertices. So let's say I'll show you 2-point rectangle. So click on that. So if you click on, this will be your one vertex and you'll have to mention the other vertex. And once this is done, you will be able to draw a rectangle. So you want to change, if you want to change the dimensions, you can type in the dimensions in your text box. So if you want, let's say the width to be say 100 and the breadth to be 40, just type in 40. And if you want to change the width, you'll have to click on tab. So tab will help you to change from your breadth to your length. And here you can type in your dimensions, say 100, something like this. And once it's done, just click on enter or left click on your mouse. So if you have any doubts, you can unmute and ask me. That's how you create. So you have other options. So line command. So if you click on line, it will ask you to enter your start point and end point. So let's say we want to draw if you want to draw a line at an angle, you'll have to mention the length of the line, let's say 100. And then to mention the angle, again, you'll have to use your tab option. So if you click on tab, it will come to your degree option, and here you can type in your degree, let's say 45 degrees. Once you do that, just click on enter, and we have a line of length 100 and at an angle of 45 degrees. So these are some of the shapes we can do. There are other shapes like we have an arc option. So if you want to draw an arc, I'll recommend you guys to like just explore these options: ellipse, arc, polygon. But the most frequently used options will be your circle, line, rectangle, and uh, and arc up to some extent, but not much. So this is your, and let's say now if I'm done with my sketch, and this is my sketch and I'm happy with the sketch, and if I want to finish my sketch, then just click on this green tick option which says finish sketch. So when I click on that, it will go back to my previous 3D screen which I told. But if I say let's say I made some error in my sketch and the dimensions are not right and I want to change it under so this is your sketches option on the left hand side, and under that you can just expand this sketches option using this arrow and double click on your sketch one. So if you double click on your sketch one, it will go back to your sketch. And here, let's say my circles diameter, I didn't want it to be 60, I wanted it to be 100. Just then double click on your dimension, which is given here, and just change your sketch dimension. So you can do that. Also, if you on the right hand top corner, here you have your home option. This is your home option. So if I click on home, this is the view in which you'll be seeing your model. And if I want any specific site to be seen, then I can click on front, top, right. And if I want to rotate it, I can click on this corner part of the cube and just move it. So that will help me to rotate along my rotate the sketch in my view. And if I want it back to my home, just click on your home option here. So that is one way. And let's say in this view only I want to move around, and this is very helpful for people not using your mouse. Here at the bottom there are few options. So if I use pan, it helps me to move across my screen like this. Or if I use my orbit option, it'll help me to rotate my sketch. And in any case, if I want to get out of these options, I can just use escape. If you have any doubts till now, you can unmute and ask. Because the chat is not loading for me, so I'm not able to read your chats.
How did we remove that grid?
Which grid? That grid is, so you're asking this grid, right?
Yes.
On the top right hand side, you have a finish sketch option here. So if you just click on that, it will come out of your grid.
I'm doing, but after that also the grid is being shown over here.
Uh, are these options being shown like solid surface, all these options on the top? Or is it still in your 2D uh,
No, no,
like
all the
option?
options are there.
Okay, then I don't think grid is an issue. Like that won't affect much. It's fine. It depends on
Oh,
the version also. So probably
thank
your,
you.
yeah. Okay, so also if you this is bottom here, like bottom of your screen, you will be seeing something called as a timeline. So this is your timeline. So you can as well go to your sketch using the timeline. So when you are using Fusion and you have multiple sketches and multiple things which you have done, the timeline is very helpful because it will show you the order in which you have done your steps. And if you want to change any of those, you can refer back to your timeline and change it. Also, So in your 2D options itself, there are few more important options like your mirror. So mirror helps you to mirror any particular object, or you can mirror is option is available in 3D as well as 2D. So if you want to mirror a sketch, you can use your 2D mirror, and if you want to mirror a body which you have a 3D model, then you can use a 3D mirror. So we'll look into that. So click on mirror, and let's say I want to mirror this rectangle. So before doing that, I'll have to draw a mirror line. So let's just draw a line like this. So I want to mirror my rectangle about this center line. So go to mirror, select your rectangle. So you will have to select the sides of your rectangle as your object. And for mirror line, click on select option and select your mirror line. Let's see this. And once you click done, this rectangle will be mirrored about this line. So depending on the line or the angle at which you want to mirror it, you can draw a mirror line at that angle and select the object. It will mirror. And let's say, so also there are some instances where I draw a shape, but the dimensions are not visible and I want to change any of the dimensions. Then I can use something called sketch dimension here. And if I say I want to change the angle between two lines, what I can do is just, so if I click on the two lines, it will show me the angle. So that is your sketch dimension option. So let's say, let me draw a line like this and another line like this. And let's say I want to change the angle between these two lines. What I can do is, there's a keyboard shortcut also. Just click on D. If I click on D, that goes to sketch dimension, or else go to create and sketch dimension here. And just select these two lines, and it will show you the angle between that. Double click on the angle and you can make it to the desired angle. Let's say I want it to be 60 degrees. It will move the lines accordingly so that your angle is 60. So if you guys are using a mouse, instead of pan option, what you can do is just click on your middle scroll button of the mouse and drag it. So that will help you to move across the screen. That okay, so that is your sketch dimension. That's also very useful. Then we have few more options. Now I think under create, another useful option is your rectangular pattern or circular pattern. Both do the same job, but in one will do it in using under like a circle, and one along a line. So let's say your we'll do a rectangular pattern for now. Click on rectangular pattern, then it'll ask you to select the object. So select, let's say we want to duplicate this circle along a specific line. So what I can do, select my circle as the object, and I can move it using these arrows along any direction. So that is, and I can change the quantity here. So we'll make it 4. And I can increase the distance here. So here, this is your quantity option and this is your distance option. So in changing these two, you can move the duplicated objects along a line. And let's say I want it across both my horizontally and vertically duplicated. So I can move this up or arrow as well, so that will duplicate along the vertical direction. So here also same quantity and distance options are available. So you can change accordingly, and you can make it symmetric and or in one direction. So that is your choice. So this is your rectangular option, and once you're satisfied with that, just click on OK. For now, we'll not do that. So that is your. So something similar is happens when you use circular pattern, but it will happen along a circular manner. Like you'll have to specify the radius of the circle, and it will duplicate along that. Then we'll so under create, I think these are the major options. Under so then we'll go to modify. So under modify we have options like fillet, chamfer, trim, extend, break, move, extract, change parameters, etc. So let's say we want to move a specific sketch or copy it. You can use the move option or click on M on your keyboard. So both do the same job. So just click on your sketch, and if you move it, you will be able to move the sketch. But I'll recommend you guys to not use this in your sketch, and you have a move and copy option in your 3D as well. Like once you have modeled a sketch into a 3D model, you can do that later on. So don't move the sketch now. Then you have a trim option. So let's say there are some parts of my rectangle which are intersecting the circle, and I don't want this part. I want to like basically remove this. You can use your trim option and just delete the parts. So basically trim is nothing but a delete option. So so to get out of any option, you have to just click on escape. That is so that trim is a useful option. Um, so there are a few more options like fillet that we'll look into during 3D part. And we have an offset option. So let's say if I want to offset a specific object, like it basically duplicates the object and offsets it. So I can click on the object and just move it. So something like that. I can give my offset distance. So if I want to offset it on the other side, also I can give my. So you can just offset it like that. So that's your offset option. And change parameter. So what you guys can do is you can create a parameter and dimension every shape using that parameter. So that in case you if you change the value of the parameter, all your shapes values will change, and you don't need to go to each shape and change its value. So parameter is also a very useful option. So using when you're doing very complicated sketches, using parameters will be helpful. So I'll show you a demonstration of that. So just go to modify, change parameter. Here you can go to your plus option here, and it'll ask you to add a new parameter. So let's say the name of the parameter is A, and you can change your unit if you want, but usually let it be in mm, and enter the value of the parameter, say 10. So so A is basically a variable storing a value 10, and then click OK. So here under user parameters, you'll have a parameter name and expression. Click on OK. Now, if let's say I draw a circle, and here let me here instead of giving the value in numbers, I'll give it in terms of parameters. So if I want it to be 60 millimeter, I can do 6 times A because A's value is 10. So I'll write 6 into A. So once I do that, just click on enter. So here we have a circle whose diameter is 60, but it's given if I want to change it, but the value is in terms of parameter. So let's say I if I change my parameter value here, instead of 10, let's make it 20. So the value of the circle will also, the diameter of the circle will change. So it will get 6 times 20, which will be 120 millimeter. So you can see the change. So that is your change parameter option. And Here, so that is all about modify options. Then we have some constraint options like horizontal, vertical, coincident, tangent, etc. So let's say we draw a line like this, but we want this line to be a tangent to this circle. So what I can do under constraints, here we have a tangent option. So just go to select tangent, select your circle and select the line. So this circle, now the line is tangential to the circle. But instead, if I click on tangent, select the line and then select my circle. So you can do in both ways. Either the line angle will change and it will get tangential to the circle, or the circle will move such that the line will remain the same, but the line will be tangential to the circle. You can do it in both ways. So that is your tangent option. So similarly, we have other options like, so let's say we draw two lines. So the shortcut for drawing a line is L, and shortcut for drawing a circle is C. And now let's say I want both the lines to be parallel. So select your parallel option, select the two lines. So now they're parallel to each other. So similarly, I can do something, let's say perpendicular. Select, but okay, we'll draw. So if I go to my perpendicular and select, so this line will move in such a way that it's perpendicular. So these options are also useful. So same way we have some like options like concentric. So in case we draw two circles but we want the center to be the same, use your concentric option. Or if you draw two lines and you want them to be of the same length, you can use your equal option. Coincident will basically make them intersect, and horizontal and vertical is basically something similar to perpendicular option itself. So any doubts till now? You can unmute and ask me. If there are no doubts, then now I'll go to the 3D part. So basically in 2D, these are the major options which I mentioned. So under create, we have a line, rectangle, circle, some shapes, and we have our mirror and some circular pattern, rectangular pattern, and sketch dimensions. So they are the major options. Under modify, we have fillet. Fillet will be looking into in the 3D part. Then we have a chamfer also will look into the 3D part. We have offset, trim, extend. So extend, let's say I want, okay, I'll show you the extend option also. So basically extend will extend a specific line. Let's say I want to extend this line until here or something. Instead of drawing, since we don't know the angle of this line, just use your extend options. Click on extend, select the line and extend it till here. So the red part shows the extended part. So it will show you till where the line will get extended. So if I am satisfied with the length of the extension, just left click on your mouse or just click enter. It will be extended till there. So this is your extend option. So that is under modify. So we have your move, create copy, change parameter. And under constraints, we have many options like parallel, perpendicular, equal, tangent, coincident, concentric. Symmetry. Symmetry is also somewhat similar to mirror. Yeah, these are the major options you will be needing in while drawing a 2D sketch. So, so we always need a 2D sketch to use the 3D part, right? So okay, so now I'll show you how to draw a cylinder using Fusion. So let's say you draw a circle in your top plane. Dimension, let's say 90 millimeter is the diameter of the circle. Once I'm done with that, just click on Finish Sketch. So now we have entered the 3D options here. So here we can use our extrude option. So extrude basically converts a 2D into 3D. So I'll show you how that happens. So click on extrude, select the surface. So this surface selected, just extrude it using that arrow. And here you can type in the distance. Let's say I want to extrude it by 250. And once you're happy with the extrusion, like the length of the extrusion, here there are a few more options. Like you can choose the direction. Let's say if I want to extrude a sketch in both the directions, I can use symmetric. So here you can see in both directions it's extruded. Or if I want it on one side, or I want it in both two sides, and symmetric is the same option, mostly the same. Only thing is in symmetric, both the distances will be the same. Like you can't change. If I click on symmetric, basically 250 along the top and bottom. But if I click on two side, I can change my top or bottom distance. Like this is still 250, but I can make this different, like say 150, something like that. So that is my two side option. For now, we'll just use one side. And so here you can type in the distance. For now, taper angle we don't need, so leave that as 0. And here you have under operation, you have many things. So for now, let it be a new body. So under operation, you select new body, click OK. So now we have a cylinder. And so in your timeline, which is on the down, it shows that you first created a sketch and then you extruded it. Any doubts so far? Uh, you can unmute in case you have any doubts. Okay, so we'll go to the next option. So next we'll create another sketch. So if you want to create a sketch, go to create sketch here. Let's for now create a sketch in your front plane. So here, let's say I'm drawing a line from here like this at a random angle. So we have something like this. And let's say we want this also to be a cylinder, but we want it to be in this plane, like a cylinder in this plane. So what we can do, finish your sketch, and I'll show you how you can use your revolve option now. So under create, you have many options. So we saw the extrude option, and the shortcut key for that is E also. You can use E. Now we'll go to the revolve option. So if you click on revolve and click on your sketch, so your profile will be your sketch which you're going to revolve, and your axis, you can select your axis. Let's see, so about this axis, we want the sketch to be revolved. So here operation is directly goes to cut because a part of the sketch was embedded in your body, but we don't want that to happen. We want it to be joint, something like this. So you can change your operation from cut to join and click on OK. So here we have something like this, right? But let's say I didn't want it to be a join, and I wanted it to be a new body, and I want to revisit my revolve option. What I can do under so the timeline here will be useful now. So just double click on your revolve here, which you used. So this is the symbol for revolve. So here, just change your operation from join to new body. So basically what happens in a new body is if you go to the drop down under bodies here on your left hand side, here you have two different bodies, two individual bodies. But if I click on join, both will be under the same body. So that's the only use. Because this will be helpful in case you're going to mirror, let's say I only want to mirror this body and not this body, then it will be helpful if they are separate bodies. So now we have seen revolve and extrude. Now let's say I want to mirror this body. So I can use my mirror option under create here. So this is the mirror option. Just click on it. So it will ask you to select the object. So select your body. Then it will ask you to select your mirror plane. So we need to select a plane. So let's say I want to mirror it about my side plane because I want this to be on the other side. This will be useful especially while designing a trachea. So once this is done, here under operation, you can choose the selected, a join or new body, both do the same job, just that new body will make it as a separate body. So for now, let's just use join and click OK. So you get something like this. So this is how you use your mirror option, revolve option, and so the same thing you can now let's say I want to combine my bodies. I want all these bodies to be this as the same body. I can use my combine option here under modify. Click on combine, select the bodies, select your target body. You want these to be combined, and then select OK. So now this entire thing is a single body. So the only advantage of using a new body is you can always combine it later on in case you want to. Now let's say I want to duplicate this body multiple times. Same, you have your pattern, rectangular and circular pattern. So now I'll show you how you use circular pattern. So it will ask you to select your, under object type is your bodies, select your body. Then it will ask you to select the axis. So let's say along my uh, Z axis. And I can increase the quantity, or let me just change my axis to so here you can just do it. So in case, so circular pattern would be along a circle, and rectangular pattern would be something like this. You can change your the quantity, the distance between the offset objects, and once you are satisfied, just click on OK. That's it. Something like this. So you get something like this. So that's how you use your pattern option. Or you can use your mirror option as I mentioned. So mirror also is the same thing. You can mirror an object along a particular axis or a particular line, anything. So now if you see in my 3D view, my sketches are not seen. But if I want to enable, like I want to see my sketches as well, just go to your drop down and unhide the sketches here. So now if you see, here you can see your sketch. It is visible. And let's say I don't want to, for now, I don't want to see this body. I can just hide the body here under bodies. So something like that. Okay, so that is about extrude, revolve, mirror, pattern, So we have few more options like cylinder. So cylinder option here will directly help you to draw a cylinder. So you'll have to just select a plane and you'll have to specify the diameter of a circle and the height of the cylinder. So that will draw a direct cylinder. So you can do that, or you can create a sketch and do the cylinder how I showed it. Or you have your sphere option here. So sphere is also the same thing. You'll have to select a center point and specify the diameter of the sphere. So sphere will be formed. So if you're in case you're using like screws, you draw a screw and you want to thread it. In that case, your thread option will be useful. But for designing the trachea, all this is not needed. So I'm not going into that. So that is mainly under create. So we'll go to modify. In modify also okay, I'll show you how you use the fillet option. So let's say I want to smoothen this edge, right? It's a little sharp and I want it to smooth it. I can just click on fillet, select my edge here, and I can specify the diameter. I mean the chamfer distance here. So let's say I want it by 10. So it will fillet by 10. But if I want it, let's say by 2 or something, just a small fillet, then I can specify here the distance and click on OK. So here this is fillet, and I'll show you chamfer also. So let me want if I want to chamfer, let's say this edge. So go to modify, chamfer, select your edge and specify the chamfer distance. So let's say 2 again. So it does the same job. So the only difference in fillet and chamfer is fillet makes it round and smoother on the edge. Chamfer basically, if you see, it moves inside at an angle. Like it doesn't smoothen the edge. It basically extrudes at an angle of 45 degrees. So that is the main use of chamfer fillet. Uh, then we have a shell option. Let's say I want this body to be hollow, then I can use my shell option. So you just click on shell, select your body. So here it'll ask you to select your face or body. Let's say the full body, and it'll ask me my inside thickness. Say 10. So it won't be visible. Let me select a face. Let's say this face, and let me just mention my. Okay, so the body is basically already shelled. So if I go to my cross section and see, like you here, you can go to inspect and go to sectional analysis and select a plane. So here, if you see, my body is hollow inside. It's basically shelled inside. You can see this is the inside part of your. So this is also pretty useful. So go to inspect and sectional analysis, and you can see your section. So that is shell. Mm, I showed you combine. So yeah, here I'll show you move and copy. So let's say I use this option. So it'll ask me to select the body, and here I can move it along any direction. Doesn't matter. So once I have moved it along any direction, and I can even rotate it. So so here you can just specify X, Y, Z distance and angle, and once you are satisfied, just click on OK. That is your move and copy option. Um, then we saw change parameters already, and another thing is if I want to change the material of my body or my face, I can use my physical material option. So here you have many options like you can just click on specific like ceramic or liquid or something like that, or glass. So if let's say I want to put this as my material to this body, just drag the hand and move it on your body. So that will give me my glass texture, or if I want a bronze texture or something like this. So it's coming like this because my inside is hollow, right? I used my shell option and I made it hollow, so it's something like this. Or if I want to, so you can change your color, whatever you want as you desire. And once you're happy, just click on close. So modify, I think we have covered most of the options in modify also. Any doubts till now? Okay, if there are no doubts. So you can see your timeline here. Now your timeline has slowly increased. It shows you each and every step. So first we created a sketch. We extruded the sketch. We created another sketch. We revolved that sketch. We mirrored it. Then we combined the objects and we used the circular, I mean rectangular pattern option. Then we chamfered and filleted these parts. Then we shelled it. So in case I want to revisit any of these previously done options, just double click on that and you can change your, you can, if you want to delete, let's say I don't want to shell it, then right click on that and click on delete. If I do that, then that particular operation which I did on my 3D model will be deleted. Um, and as I mentioned earlier, if I want to, so since this is a 3D model and if I want to inspect how it looks from inside, like I want the inner view of it, just go to inspect and sectional analysis and select any plane in which through which you want to cut it. So, and if you move using your cube option here under home, and if I let's say move my, I can move my cut plane. So if I want to cut it fully, if I want a specific part. So this is very helpful. And let's say I just want to change, let's say this is the plane in which I want my sectional analysis. Just click on OK. So this will be my sectional view. And let's say I want to hide my view. How do I do that? I want my 3D object back and I don't want this to be a sectional view. I go to analysis and under analysis, I just click on the eye option again to hide the section. So I get my 3D view back. So that is my sectional analysis. Now we go to construct. So here you have a construct option and here you have many options. Uh, so mostly will be like most useful ones for your offset plane, your mid plane, and your tangent plane. Mostly we'll be dealing with these. In case you need other planes, also, I mean, you can try out other options, but we'll be looking into these mainly. So let's say I drew a sphere. Okay, so we'll create a sphere for now. And let me just, for now, let me hide my body. So let's say this is my sphere and click on OK. Now I want to draw something which is tangential. So I can use my tangent plane option. But we can even use tangent to a surface. So since a sphere is completely round, the tangent won't work here. So let me, let's say using this body, I can use my tangent to this surface. So what I can do, construct tangent plane, select your surface. Now we have got a tangent plane. So I can change, let's say I want it to be tangential like here, anywhere. Let's say here. So once I'm happy, I can change my angle here, let's say by 90 degrees. Then click OK. Now we have a plane here and we can even create a sketch on this. So how we create a sketch? We select the plane, go to create sketch. Now let's say let me draw some circle of some distance here. Once I'm happy, just click on OK. Unhide your sketches so that the sketch is seen. Click on that and extrude. Click OK. So now you see there's a hole in your cylinder. So that's how you use your tangent plane option. Then we have offset plane option. Let's say I want to draw a sketch at at let's say some height from a plane, or I can use my offset plane option. So to do that, click on offset plane, select a plane. So let's say I want to offset my top plane at a certain distance. Select your top plane and move it up by the desired distance. Let's say till here. And once you move it, just click on OK. So now I can create a sketch in that. So select your plane, create sketch, and draw a circle. Now you can draw a circle like this and click on finish sketch, and then select the surface. You can extrude it a little, something like this. So as I mentioned earlier, if you're designing a trachea on Fusion, you'll have to assume everything to be perfect cylinders, even the rings of cartilage. You can change the distance, you can change the diameter of the cylinders, but only, I mean, if you want to add irregularities to the cylinder, you will have to use blender, and we'll see that in the next class. So let's say now since I have this body and I want this body to like, I want multiple copies of this way. So what I can do now, I can use my pattern option, rectangular pattern, select your body and select the axis, let's say axis. So I can just increase the quantity, and I can choose the distance between each. Once I'm happy, I'll just click on OK. So this is how it will look. So any doubts so far? Okay. Okay, then now we'll see how. So if I want to construct a plane which is offset to this plane, I again click on offset plane, and instead of selecting my default top left side planes, I'll select this surface, and I can move it as, let's say, at 25 distance. So I can get a plane here, and we can create a sketch in this plane also. So we can draw a circle, finish your sketch, and select and extrude. Something like this. And again, I can create multiple copies of this. So I'll just go to pattern, rectangular pattern, select it. To select the axis, this time I'll select this line. So you'll have to enable your sketch to be seen and select this line so that it will get pattern along this. Once that is done, you just move it, change. Something like this, and then click OK. So you can do that. Then you can as well mirror all this. So what you can do, click on mirror, select your bodies, select the mirror plane. So this will be your side plane, and then click OK. So you can mirror it. So it's advisable to do one side and just mirror it. But as I mentioned earlier, you can change the ring sizes. You can change the, if you don't mirror it, you can even change the length of this bigger cylinders. Like you can change this length, make it longer. So if you want to do that, just click on the edge and just extrude it. Something like this. So you can do that. Uh, yeah, this is mainly about how you mirror and extrude. So if you click on the play option under your timeline, it will show you how we started and what we did. So if I want to revisit any specific part, just pull this back in your timeline like this. So it will show you that part, or and you can change things later on also. That's the only advantage of Fusion, but that is not there in Blender. So in Blender, you don't have a timeline. So if you want to change anything which you did earlier, you will only have to use your Control Z option. But in Fusion, you have the liberty to change anything later on. So you can even do plane at an angle. So let's say we have a, let me hide my body still for now. And let me draw a rectangle. So let's say I want to I want to plane at an angle. So here what I can do, construct a plane at an angle, select your line and now rotate it. So here you get a plane at an angle. So you can specify your angle here. And once you're satisfied, just click on OK. In case you want to construct a mid plane, how you do that? Select mid plane, select the two surfaces through which you want to mid plane. So let's say my this surface and other side, so this surface. So So if you select the two surfaces, you get a plane which is passing through in between the two surfaces and cutting the surface equally. Once you are happy with that, just click OK. So this is mainly what you will need for designing a trachea. You can add your creativity, you can change the colors, you can change the ring size, you can change the length of the cylinder, the thickness. So I have explained most of the more common options in Fusion, and I think this is all you need for designing a trachea. Any doubts till now, you can ask me. So I am done with my part.
I was only able to make a single cylinder. It's very confusing.
Uh, yeah, a fusion is a little confusing. Like you'll have to practice it a lot to get a hang of every option. So in case you have any doubts regarding the options, you can just see our recorded session again. And so
So I think you understood her explanation well. So you need to model only the central part of the trachea, not the bronchi, not the larynx as well, just the central part with the rings of cartilage. Did you get it? So some of you have asked the pictures of lab grown organs. I'll present it. I'll present it now. 1 minute. Okay. So I'll show it now. Please wait for 2 minutes. I'll be back. Okay, guys, let me present my screen now. So can you see this part which I'm circling with my cursor? This middle part, only this, you need to model only this part. No need to include the larynx and these bronchi. So because this is much simpler for beginners, that's why. Did you get it? So
Yes.
some of you asked, okay, some of you asked the pics of lab grown organs. So the this pic is nothing but a brain organoid with two eyes in it. And this pic, the B pic is nothing but the extracellular matrix after removing all the cells from the heart. So these are bioprinted organs. So these are sensory organs, right? And this is the first bioprinted heart. I hope you got it. If you want more resources on it, I'll share it to you in the homework or in the resources channel. So let's have a sneak peek for next session, which is the success stories of the lab grown organs, and we'll be discussing the challenges which we face with organoids and lab grown organs as well. And finally, we'll be concluding this module with by discussing the ethical considerations as well. So that's it for today, guys. And I'll be sharing these resources with you all. You can learn more about Fusion 360 with these two things as well. So thank you for attending this session. I hope you found it exciting. And if you have any doubts, you if you have any doubts, you can stay back, or else you are welcome to leave the session. Can we bioprint an entire face? Okay. You can bioprint the face, but like you can just print the skin of it. Like I'm not very sure about it, but face in the sense you are asking just the skin part, right? We can print that. So we'll be discussing all these things in tomorrow's success stories. So thank you, everyone. If you have any doubts, you can stay back, or else you're welcome to leave the session. And some of you have asked doubts regarding the software as well, about how to download it. If you have any doubts, you can stay back and you can ask Deeksha.